this video lecture is going to be over Chapter 7 and Chapter 8. So we'll start first with Chapter 7. Chapter 7 talks about sex determination and sex chromosomes. So the two modes of sex determination that were early described by Wilson were modes of sexual differentiation identified in insects. So the two modes were named after the model organisms that they were first detected in. So Protiner, after a butterfly species. And in the Protiner mode of sex determination, what we find is that you have an XX and an XO, meaning one sex is homogametic. They have two copies of a sex chromosome. And in the other sex, in this case the males, they're heterogametic, meaning they have one copy of a sex chromosome. So homo, same, hetero, different. And what we find is that what determines the overall sex of that offspring is whether they have the two or the one chromosome. So in females, having two will develop into females. Males, because they only have a single X chromosome, don't develop into females and develop into the male themselves. So these the gametes that get formed from these individuals. So when you have a female and she creates gametes, she's going to have two gametes, she'll create four gametes that they'll all have. In this case, in the Protiner butterfly, they're going to have 6 autosomes. That's haploid. And they'll have one X chromosome each. But in males, because they only have one, there's two potential gametes that get made. One that has the X and the other that doesn't. And so fertilization with the gamete that doesn't have the X would result in a male. Fertilization with the male gamete that does have the X results in a female. The other mode is named after the milkweed. So it's the Ligeus mode of sex determination. In the Ligeus mode, we have also homogametic and heterogametic, but this time the heterogametic has a different chromosome. So this was named a Y chromosome. So you have an X and a Y, and this is similar to what we have in humans, right? We follow the Ligeus mode of sex determination, in which males are heterogametic with a Y chromosome, and genes on that Y chromosome are what differentiate the male phenotype. Now, this isn't the only mode of sex determination in organisms. We also have other genetic sex determination systems. And when I say genetic, I mean that there's actually a chromosomal determining factor that leads to sex. Those systems could include what we call a ZW system of sex determination. This is similar to the XY, but the genders are swapped. So the homogametic is actually the males in the ZW, and the females are the heterogametic organism where they have a Z and a W chromosome. And this occurs in birds. It's found in a few fish, but it's less common. Fish tend to have a lot of non-genetic sex determination modes. It's found in some insects and some reptiles, but mostly in birds. And you can see here, the Z chromosome is shorter, the W chromosome is long, and that's what dictates the female. There's other non-genetic modes of sex determination, things like temperature dependent sex determination, hormonal sex determination, but we're going to just focus on the genetic mode. So you need to make sure you understand the Protiner and the Ligeus mode and the ZW. Now we're going to shift and we're going to start talking specifically about human sexual determination and differentiation. So in humans, the Y chromosome is responsible for sex determination of the male. If we look at the Y chromosome, there's actually not that many genes on the Y chromosome. There's between about 30 to 200 genes, something like that, as opposed to thousands of genes on the X chromosome. If we look at the chromosome itself, we can divide it into regions. Those regions are the PAR regions that are found on either end, and the MSY region. So this PAR stands for pseudo-autosomal regions. And these are the regions of the chromosome that actually share homology with the X chromosome. So this is the regions that match the X, so that during meiosis and mitosis, that they, really during meiosis, in which they will align together and form our homologous chromosome pairs. Okay. There's no coding genes in these regions. It's mostly on the telomere, so there's not really any genetic recombination that occurs here. The MSY region stands for the male-specific region, and within this we can break the material up into what we call euchromatin and heterochromatin. So the euchromatin regions contains functional genes. These are genes that will be turned into proteins. The heterochromatin, sometimes people call it junk DNA. It's not really junk DNA. It's just non-coding. They play an important role in some gene expression, but there's no proteins themselves that are coded within this region. Within the euchromatin area, the most important region is this SRY region. This stands for the sex-determining region on the Y chromosome. Really is a fairly recent discovery. It wasn't discovered until 1990. And this is the region that contains the gene. The gene is the testes determining factor. It's a protein that triggers the testes to form during embryonic development. If we look at that embryonic development, an embryo at around 7 weeks has what we call bipotential gonads, meaning it has the precursors that could become both testes or ovaries. Those are the Wolffian ducts and the Mullerian ducts. Now, if an individual has a Y chromosome, then around 7 weeks, we start producing that TDF. And that TDF causes the Wolferian ducts to start differentiating and growing and dividing. That protein binds to receptors. It causes it causes those cells to start developing. Those cells, and don't worry, this is more information than you need to know. I'm just going in because it's interesting, but those cells start producing compounds in themselves. So the Sertoli cells in the testes start producing what's known as anti-Mullerian hormone, which causes the Mullerian ducts to atrophy and disappear. So we lose the female genital tract. The Leydig cells in the testes start producing testosterone, and that continues to stimulate the growth of the Wolferian ducts. And then our germ cells will start producing the spermatogonia, the sperm. If we don't have a Y chromosome, then what we see is when we hit that 7-week mark, we don't start producing the TDF. And so by around weeks 8 to 9, the Mullerian ducts, they actually start to differentiate on their own and start to become the fallopian tubes and the ovaries, and they develop into the primary sexual characteristics, the primary sexual organs of the female. Likewise, as these develop, the Wolffian ducts are going to start to atrophy. There's no hormones that are required for this to start growing, unlike, say, the testes that needs that testosterone and that TDF. In our previous chapter, we spoke about non-disjunction, about how we can have a non-disjunction event during meiosis in which either homologous chromosome pairs fail to segregate or sister chromatids in meiosis 2 fail to segregate, and that can lead to an improper number of chromosomes. So let's talk about what this can do with just the sex chromosome. We'll talk in Chapter 8 about other chromosomes, but in sex chromosomes, there's a number of conditions in which this can occur, and it's tolerated better than, say, in an autosome. The first that we want to talk about is poly-X, where we have more than 2 X's. Generally, can be 47 chromosomes instead of 46. You can even have more. But the presence of 3 chromosomes with a normal number of autosomes results in we're still going to differentiate and be female. However, some of the secondary sexual characteristics sometimes become underdeveloped. And by secondary sexual characteristics, what we mean are things like the mammary glands, the breast tissue, some of the other characteristics that we associate that come on during puberty. This is actually fairly common because you can result in this by having an X-ray. You can see if we go to these non-disjunction events, it can occur here in 50% of the gametes when we have a non-disjunction event in meiosis 1, as well as when we have a non-disjunction event in meiosis 2, it can still occur in 1/4. So about 1 in every 1,000 females is actually born poly-X. Poly-X in males, where we have 2 X's and a Y, also fairly common. The frequency is about 1 in every 500 to 600 males, so more common than poly-X. We call this Klinefelter syndrome. And there's a couple of characteristics that kind of differentiate individuals that have this. For instance, they're normally tall, but they also have very long arms and legs. There can be a underdeveloped testes, prostate gland. Sometimes they won't be able to grow a beard. That would be part of the secondary sexual characteristics. They are phenotypically male, so they're going to look mostly male. But if you have Klinefelter, generally you're infertile. Sometimes you can actually start to have what's called gynecomastia or breast development that occurs during puberty. But generally, you're going to look phenotypically like a male. And it can manifest itself where you can have 47, where you have 2 X's, 3 X's, 4 X's, and so on. Poly-Y in males, less common than Klinefelter's. It's similar to what we see in poly-X, so we have about 1 in every 1,000 males will be poly-Y. There's not really any consistently shared characteristics between individuals other than males tend to be over 6 feet tall. In some people, it's documented that there's subnormal intelligence, some mental handicap, but not in everybody. Most people aren't really affected by this. There's not that many genes on the Y chromosome, so having an extra copy really doesn't cause that many problems. What happens if we only have an X and we don't have a Y? So we're XO. This is what we call Turner syndrome. So in Turner syndrome, phenotypically, you're going to be female. You're going to look female. This affects about 1 in every 2,000 females that are born will be born with Turner syndrome. There are some general characteristics. It's kind of hard to associate. It's not like you're going to be able to see somebody and be like, oh, they have Turner syndrome. But some of the things that have been documented is generally decreased height. They may have what kind of like some malformed features, like a webbed neck. That just means that they have a thicker neck at the base. They could have a high palate. There's been documented, heart conditions, kidney conditions, infertility can occur. Not always, but it can occur. Okay, but phenotypically female. Now, this brings us to a unique concept of what we dub dosage compensation. So, in individuals, like females that have 2 X's, one of the questions that's posed is, okay, if a woman has 2 X's and a male only has 1 X, then do they produce different amount of proteins from the genes associated on the X chromosome? And what we see is rather than having excessive expression of X-linked genes in humans and other mammals, that's one way you could compensate, right? In males, the X could produce twice as many transcripts. It could overexpress to make up for the absence of the X. We don't see that in humans and other mammals. Instead, what we see is that we have dosage compensation in which we decrease the production of proteins on one of the X chromosomes in females. Because we have these two means by which we can do it. We could we could increase the expression of that X. So here in males, you can see if this is what females have, we could increase it in a male, have a twofold transcription of that single X versus in the female, and then we'd be okay. Or we can inactivate one of the X's. And that's what happens in humans. So in humans, one of the X's in a female will become inactivated and turned into what's called a bar body. Now, this is random as to which X it is. It could be either the maternal or the paternal chromosome that gets inactivated. And it gets controlled by a number of transcripts that they get produced on the X chromosome. The main one is called the XIST, the exist gene. It gets turned on early in development when cells are dividing during embryonic development. And we start transcribing that exist gene. It doesn't code for a protein, but it makes an mRNA. And that mRNA, it binds to another region on the X chromosome, and it basically wraps it up and causes methylation to occur, and it doesn't allow that DNA to be accessed by transcription factors to transcribe the proteins on it. So you can see that right here in a cell. Here's the bar body. So this would be an X, right? An X and an X. Or your X is somewhere in here, but then the bar body gets all bundled up, has a bunch of histones associated with it. Now, if we have inactivation of one of the X chromosomes, then one question we have is why is Turner syndrome, so when we only have a single X, why are they not entirely normal? Because you only need one X. So if a female is only born with one X, why does it matter if she only needs one? Or when you have females with triple or tetra X's, why are they not normal? Why were some of these things that we're talking about not quite right? And some of the answers or possible explanations, we have hypotheses. We don't know for sure. We have some reasonably good hypotheses that have some support, evidence that supports them. One is that inactivation doesn't take place in the early stages of development of the gonadal tissue. So perhaps when we have a failure of this to occur early during the gonadal tissue, that's what results in different people experiencing slightly different phenotypic characteristics when they have Turners or Klinefelters Turners or poly-X, right? The other thing is that maybe not all the X chromosomes get inactivated. So if you have, extra X's, maybe you don't produce all the bar bodies. Maybe in some individuals they do, and there might be something going on that in one individual that is experiencing more side effects of being poly-X than another. Now we know that X inactivation is random because we have studied this and tested this and made observations. And one of the first observations about that it could be random was dubbed the lion hypothesis by the individual who studied it. And they basically said that these somatic cells early during embryonic development would randomly inactivate some of the X's, and we can see those patterns. So like a calico cat, the hair coloring, that's a gene that's found on the X chromosome. And so early, when this cat was just a bundle of cells and they were dividing, you started getting cell lineages that didn't have those X's being inactivated. And at some point you inactivate some, and you get genes that are on the maternal or paternal side that could lead to different coloring. So you can get the calico that occurs in males, where you're XXY. And then you can get the heterozygous, like the turtle shell, coloring in females where it's, a codominance is occurring, and you can see the actual different cell lines. Like these cells stem from one area, those cells stem from another area. Then the X's were inactivated. When they divided, obviously you only replicated the X that wasn't inactivated. The other one doesn't get copied and replicated. Okay, that finishes it for Chapter 7. So again, make sure you know Legeus, Protner, dosage compensation, Klinefelter's-Turner syndrome, poly-X, poly-Y, what those kind of what those means, how those can occur from non-disjunction. Chapter 8 talks about chromosomal mutations that aren't sex linked, right? They're not on the X chromosome. So while most species are diploid, meaning they have 2 copies of a chromosome, and they only contain, two chromosomal sets, there are some variations in which this gets violated. Sometimes you get a change in the total number of chromosomes that exist. If the total number, so in humans, if we're 46, we could have it. It's possible that somebody could have an event occur in which they get a whole other set of chromosomes. It's probably not viable. That person probably won't live. But that could happen. We could have a change in the total number of chromosomes. We could also have changes that occur in just the genes found on a chromosome. So we could have a section of a chromosome either deleted or duplicated. Or we could have those regions rearranged where the gene order gets turned. All of those changes are what we call chromosome mutations or chromosome aberrations. And we use that, the term aberration really to distinguish it from gene mutations. So these are things that are happening on the chromosomal level. We've already talked about one type of chromosomal aberration, non-disjunction. Non-disjunction can lead to us having an uneven number of chromosomes. We can get what's either called a trisomy, where we have 3 copies of a chromosome once once fertilization occurs. So in this case, 2 from the mother, 1 from the father. Or we could have what's called a monosomy, in which we have 1. Okay. Now, the loss of 1 chromosome produces a monosomy in humans. This has severe phenotypic effects. Most monosomies will never develop and never be born. We talked about some monosomies on the X chromosome and why those tend to be tolerated because of X inactivation, and you can exist with 1 chromosome. But for most other autosomes, it really doesn't happen very often in humans or animals. Trisomies, where we have an extra copy of a chromosome, those do occur as well, and some of them are tolerated. Most of them are lethal, but there's a few. Sex chromosomes obviously have a less dramatic impact on the phenotype than if it was on an autosome. But Down syndrome is an example in which there's a couple ways this can occur, but you get an extra copy of the 21st chromosome. So it's a trisomy 21, and that can have phenotypic effects. Other ones, Patau syndrome, that's chromosome 13, or Edwards syndrome is chromosome 18. Those are all trisomies. They'll survive to term, but they're going to have extreme phenotypic effects because, again, we have an unbalanced. If you have an extra chromosome, you're producing more transcripts, more proteins than normal, and that can cause problems. Additionally, it also can cause some problems when you start doing mitosis and cells are dividing and things aren't necessarily, or meiosis for that matter, when they're dividing, you don't have equal pairs. Why do we know that most of these things aren't tolerated, trisomies and monosomies? Well, it's perceived that, about 20% of all conceptions, so when a man and a woman conceive a child and fertilize an embryo, about 20% of them end up resulting in spontaneous abortions, or what people commonly call miscarriage. And when we've looked at, when doctors and researchers have looked at miscarried fetuses, what they find is that about 30% of all of those miscarriages occur because of chromosomal imbalances. And so sometimes, I know it's a miscarriage is always a difficult thing to go through, and there's sadness that goes along with it, but there's also something to be said for the fact that that chromosomal imbalances generally aren't tolerated well. The few that are have extreme phenotypic effects that can that you're going to have lots of different problems with it. So the body has a mechanism in which it recognizes this is not going to yield a viable fetus, and so these things occur. Monosomies generally aren't found in abortive fetuses, and we can calculate what the probability of a monosomie occurring or trisomie. So what that suggests is that monosomic gametes, they may be functionally impaired. So a sperm or an egg that only has a single chromosome, it may not be able to swim or to become fertilized altogether. Otherwise, we would see these in those spontaneous fetuses. Okay, so moving on. Monosomies, trisomies, that's just for single chromosomes. What if we have all of our chromosomes, an extra copy of all of them? We call that polyploidy. Okay, the name of polyploids is based on the number of sets. So we are diploid, that means 2 sets. You could be triploid, in which you have 3 sets, tetraploid, pentaploid, so forth. There's a couple ways that polyploidy can originate. One is that you can have an additional of 1 or more sets of chromosomes that become identical from the same species. So that would be autopolyploidy. Maybe there's a replication or a non-disjunction event for all of the chromosomes. That would be autopolyploidy. The other thing that could happen is that you could have a mating between 2 species that have different numbers of chromosomes. So that would be allopolyploidy, in which, right, here's this species, so you have these chromosomes, these species have some different chromosomes, they reproduce, and now you get a tetraploid. Now, polyploidy, whether it's auto or allopolyploidy, generally isn't tolerated very well in animals and invertebrates because of our developmental genetics that we go through. But in plants, it's tolerated actually quite well. So lots of plants are tetraploid. You have a lot of allopolyploidy, in which plants from different species. That's why you have all these weird citrus fruits, right? Where you take like a tangelo, where it's a tangerine, and a pomelo that then cross-pollinate and you generate these types of fruits and stuff. Those are allopolyploids. Other variations that can occur, okay, besides just additions. We can have rearrangements of our chromosome segments. So the 4 types of things that we can have is deletions, duplications, inversions, and then translocations. Okay, 2 of these are going to result in either a loss or a gain. That's going to be deletion and duplications. Okay, inversions and translocations, we don't lose any genes, we don't gain any genes, but we're just changing the order of those genes on the chromosome. So let's start with a deletion. So I just told you, 2 ways. Total amount of genetic information changes through deletions, duplications. Remains the same but rearranged through an inversion and translocation. In a deletion, we have a missing region of the chromosome. It breaks in 1 or more places, and that portion gets lost. That results in what we call a deletion or a deficiency. These could be at the terminal end. They could be in the middle. When they're in the middle, we call them intercalary. And generally what happens, sometimes that chromosome, it loops on itself, and then that causes, it fuses, and someone tries to fix it. I say someone. Enzymes try to fix it. They end up cutting it, and rather than ligating it back together, we lose. Maybe we ligate these ends together, and so we lose this section. And this would be an intercalary deletion. Sometimes just we have a breakage of the chromosome at the tip, where we lose just the end of it. That can result in a deletion. Sometimes during replication, right, we might have a loop, and then when we replicate, we don't copy that part. Duplications is now where we have a repeated segment of genetic material. This arises when we have an unequal crossing over during meiosis. That's how we get duplications. So instead of these two homologous chromosomes, they don't align correctly. And so now instead of having the chiasma form in the proper location, which would be here, it forms at an improper location, and it results in an unequal crossover event. So we have things where, like in this case, we have all of our genes, but here we don't have a C, so a deletion technically occurred. But we have a duplication here. We have two Cs, right? So that's a duplication. This can lead to gene redundancy. In fact, we know this happens. This has happened on an evolutionary timescale in organisms. We have lots of ribosomal genes that make lots of different ribosomes, lots of cytochrome oxidase genes, which probably resulted because of gene duplications. We can also now, let's talk about our inversions and translocations. So an inversion, this causes a rearrangement of the linear sequence. So we don't lose any genes, but we can change the arrangement of it. And sometimes turning a chromosome around 180 degrees, it can have some effects on, sometimes genes are supposed to be transcribed in order. How this occurs, well, similar to a deletion, we form an intercalary loop, right? And then those gaps get created by enzymes trying to fix this loop. And then instead of ligating it correctly back together, we ligate it incorrectly with enzymes, and so we revert or invert the sequence. This can cause some interesting things when a centromere is involved. So we can have paracentric inversions. This doesn't change the relative length of the two arms of our chromosomes. So you see here, all the chromosome lengths are about, the same. But you can get, you can end up changing where the centromere is. So like we have, in this case, this one ends up with two centromeres. It's because the inversion loop, is in the middle. It doesn't contain the centromere in that loop. So you can have things in which you get something like this, where we have, two centromeres on a single chromosome. Obviously, that's going to have problems when you start to try to align things during meiosis or mitosis. If it doesn't have a centromere, it's not going to align very well. So you can see how you can get some problems from an inversion. When we have a pericentric, this includes the centromere, so it does change the relative lengths of the arms. So we're no longer, here we were like A, B, and then we had 4. And they were 3 and 3. So it doesn't result in a change in the arm lengths like we see over here, but we still retain centromeres on all chromosomes. Okay, so again, you can see it. I talked about this, how we get on the paracentric, how we can be dicentric or acentric, and how you don't get that on the other type. Last type of chromosomal aberration is a translocation. So this is now where we move a segment of a chromosome from one chromosome to another chromosome in the genome. And it's usually a non-homologous chromosome. So when we have a reciprocal translocation, that means we exchange segments between two non-homologous chromosomes. So both chromosomes exchange. In a non-reciprocal translocation, we don't have exchange on both. I'll show that one in just a second. Usually this occurs when we get a weird synapse configuration during meiosis, and we get these two patterns that can form where we could have an alternate segregation or an adjacent segregation. I'm not going to test whether you know the difference between alternate and adjacent, but just know it's a vocabulary concept. In a translocation, we may have, chromosome 1, a region from chromosome 1 gets shared with a region from, say, here, chromosome 4 or whatever. In a non-reciprocal translocation, we aren't having equal exchange. So in this case, we might just have one chromosome that's gaining apart. We call this sometimes a Robertsonian translocation. This is one way in which you can get Down syndrome. So I spoke about how Down syndrome can result from a trisomy. But another way is that you could have a breakage on the end of one of your 21st chromosomes, and it can become translocated onto the 14th chromosome. So this happens sometimes. This is generally the type of Down syndrome that we call familial Down syndrome, where you can do a pedigree chart and you see when Down syndrome runs in a family. It's because this may have happened to an individual, and then you can see that they now have a copy of their 14th chromosome that has a section of the 21st chromosome that can lead to that Down syndrome offspring, in which they're having normal. They have a copy of the 14th chromosome that has that extra bit on there. They have a normal copy of the 21st chromosome, and then when they pair with another individual that has a normal 21st and 14th, that results in this, which is essentially like a trisomy for 21. With this, you can also have carriers, right? This person it doesn't have two copies coming from the mother. It was a monosomy here, and so they don't have two copies of chromosome 21. So they're in essence normal, but they are carrier for Down syndrome because, again, it's possible when they do their gamete formation, there's a 1/4 chance they're going to create a gamete like this that will result in a trisomy and Down syndrome. Okay, so that goes through all the aberrations, polyploidy, monosomies, trisomies, and then deletion, duplications, translocations, and inversions. Make sure it's a lot of vocabulary, both for this one and the last one, that you understand it. You're going to be tested on that vocabulary.